Good morning, dear students. Today we are going to study one of the most important topic in cell biology, the cell cycle and checkpoints. For this topic, the main reference books are Albert's, that is Essential Cell Biology, Karp Cell and Molecular Biology. These books will provide you the diagrams and conceptual understanding. So I will recommend these two books, especially for the mitosis diagrams and for regulation of checkpoints. The main objective of this topic is to study the cell cycle to make you understand how cell divide in a regulated and accurate manner. So by end of this lecture, you will be able to explain what are the different phases of cell cycle, understand the checkpoints, and you will be able to describe the role of different cyclins and CDKs and their relationship between the checkpoint failures and the cancer proliferation. So why cell divide? So in today's lecture, you will learn definition of cell cycle, phases of cell cycle, checkpoints, cell cycle regulations, and their significance. So why cell divide? Cell divide for the growth, for the replication of DNA, for preparation, for divisions, and finally divide that make daughter cells again and enter into the same cycle. So every living organisms grow because cell divide continuously from a single fertilized egg. Trillions of cells are produced through the controlled cell division. So cell cycle is the sequence of events through which a cell grows, copies its DNA and divide into two daughter cells. So that today we will learn and understand. So in this illustration, you can see how the cell cycle take in action, how to make two daughter cells from one parent. So what is cell cycle? It is a sequence of events that the time a cell first arises as a result of cell division until the time when the cell itself divides into two daughter cells. So it consists a period of growth and development, a DNA replication, preparation for division and cell division. Cell after division begin a new cycle. So what is the importance of cell cycle? Cell cycle is important for several reasons. First, for the growth and development. So a single fertilized egg develops into a complete organism through repeated cell division. So it is essential for organism growth and to drive embryonic development. Second, tissue repair and regeneration. So when tissues are damaged, new cells replace the old one and injured cells. So it helps to replace the damaged or dead cells and important in wound healing. Third important parameter is to maintain the genetic stability. So it ensures accurate DNA replication and segregation to prevent the mutation. So DNA must be copied accurately to daughter cells that receive identical genetic information. And fourth, last important, it prevents the diseases. So proper regulation prevent uncontrolled cell divisions. Dysregulation leads to the disease like cancer. So when cell division is properly controlled, diseases are prevented. But when regulations fail, so uncontrolled division can lead to the cancer. So this slide explains the different events of the eukaryotic cell divisions. So cell cycle has two major phases, that is interphase and mitotic phase. So interphase is the preparation phase and mitotic phase is the actual division phase. So an important point, interphase is not a resting stage here. In this stage, the cell is metabolically highly active, but it prepares the cell for the division. So these are the two main stages of the cell cycle. This is complete interphase. Interphase is further consist G1 phase, S phase, G2 phase and M phase. So this is the M phase is known as the mitotic phase. So stages of the cell cycle divided into two major phases, interphase and the M phase. So during interphase, cell grows and make a copy of its replicate the DNA, and during mitotic phase, the cell separates its DNA into two sets and divide its cytoplasma, forming into two cells. So in M phase, there is a nuclear division and the cytoplasmic division. So together, G1 phase, S and G2 phase make the interphase. So in G1 phase is the growth phase, S phase is the DNA replication, and G2 is preparation for the mitosis. So in this slide, it is explained in detail. So growth and preparation take place in this whole interphase. M phase is a cell division. So G1 is also known as the gap one, where the growth of the what nutrients is required for the normal functionings and what is, so all the things that cell grow, carry out all normal functions that occurs in the G1 phase. S where the DNA get replicated. So DNA synthesis takes place only in the S phase. And in G2 phase, G is the second growth phase. So that is, in this phase, further growth occurs, spindle proteins are synthesized, and the cell preparation for the mitosis at the G2 checkpoint. DNA damage is checked and repaired. This acts as a final quality control step before the mitosis. So this is G1 phase, first growth phase. So what are the key events take place in the G1 phase? Rapid cell growth, synthesis of proteins and organelles, cell increase in size, and the checkpoint is this, that is known as the restriction point. So here so at the end of G1, the cell reaches an important checkpoint that is known as the restriction point. Here the cell decide whether to continue division or it should enter into the G0 phase. So the cell checks nutrients, growth signals, and DNA damage. G0 phase is resting or quiescent stage. Cell exit the cell cycle and stop dividing. So some cells enter temporary G0 and can divide again later, for example, the liver cell. But some cells enter permanent G0 and never divide again, for example, neuron cells, cardiac muscle cells. So these cells remain metabolically active but do not replicate DNA. So that is G0 phase. So this is a G0 phase when the a phase where cells exit the cell cycle and start dividing further. So G0 phase again divided into two types, temporary G0 and permanent G0. So in temporary G0 can re-enter cycle like the liver cells, and permanent G0 do not divide, that is a neuron and muscle cell. So what are the main characteristics of G0 phase? They metabolically remain active but non-proliferative. There will be no DNA replication. And importance of G0 phase is to maintain the tissue specialization and prevent unnecessary cell division. But so G0 phase, it is the resting stage phase but functional state. S phase, that is also known as the synthesis phase. So you can see this is an unduplicated chrome. So DNA get replicated during this phase and there is a formation of two duplicated chromosomes. So key events are DNA replication take place. Each chromosome form two sister chromatids and centrosome duplication take place here. So what are the important features here? DNA content get doubles. So from 2N, it becomes the 4N. So importance of this phase is that ensure that each daughter cell receive identical genetic material from the parent cell. So that's why it is known as the synthesis phase. Here the genetic material gets synthesized, duplication state. Then G2 phase. So further cell growth take place, synthesis of spindle proteins take place, and it get prepared for the mitosis. So checkpoint here is DNA damage is checked and repaired. So importance of G2 phase is that it ensure error-free DNA before it enter into the M phase. So final quality control before the mitosis. So M phase is the mitotic phase. So it includes two processes, that is mitosis karyokinesis, where the division of the nuclear content takes place, and the cytokinesis, division of cytoplasm. So mitokinesis again, that is mitosis karyokinesis, division of nucleus. So it further divided into four stages: prophase, metaphase, anaphase, and telophase. In So you can see here in prophase, chromosome get condensed, whereas in metaphase, chromosome align at the equatorial plate, and in anaphase, the sister chromatids get separated and telophase, two nuclei get reformed. So finally, cytokinesis divide the cytoplasm and produce two daughter cells. So next important topic is cell cycle checkpoints. So these cell cycle points are the surveillance system. They act as a surveillance system because they monitor the cell cycle progression. They prevent the DNA damage, incomplete replication, and the chromosome segregation error. So they function like quality control system in the factory. So that's why we are saying them checkpoints at every stage of the G1, S, G2, and M. They check whether cell should enter into the next phase or not. So that's why they prevent entry into next phase of the cell cycle and also called the DNA damage checkpoint. So checkpoints are control mechanism that ensure proper progression. It prevents the error like DNA damage, incomplete replication, and act as a quality control system. So first checkpoint is G1 checkpoint, that is also known as the restriction point checkpoint, where it is located. So you have to learn the location and their key feature. What are the main properties of those checkpoint and what things they ensure for the next phase of the cell cycle division. So location is end of G1 phase. So before the S phase, so for what they check, they check the cell size is sufficient, nutrients what are required for the replication are sufficient or not, growth factors, growth signals are sufficient. So they check for nutrient, growth factors, and DNA damage. So key mechanism is here. The protein P53 play an important role in this G1 checkpoint. So they activate the repair pathway if any error is found in the G1 phase. If damage detected, the cell cycle arrest or apoptosis take place. And if it get repaired, then the phase, it will proceed to S phase. If it is not get repaired, then it will enter into G0 phase or undergo apoptosis. So this is the most critical checkpoint for the cell phase division. Then G2 checkpoint. So at the G2 checkpoint, the cell verify. So this is at the end of the G2 phase before mitosis. So it check for the whether DNA replication is complete, whether is there any damage or not on the DNA during the replication process, and what are the key regulatory that check these points. There is a cyclin B and CDK1 that help the cell enter mitosis and to check the DNA repair enzyme. If the repair system is done, then it will allow the cell to enter into the mitosis, and if it is not able to repair, then delay the cycle for repair and prevent the entry of the damaged DNA into the mitosis. Third checkpoint is the M checkpoint, that is also known as the spindle assembly checkpoint. So this checkpoint is at the position of the metaphase of the mitosis where the spindle formation take place. So it check for the proper attachment of spindle fibers to the kinetochores. You can see here, this is a prophase during the prophase and this is the metaphase. So at metaphase, chromosome align at the metaphase place and there is a spindle assembly. So now It inhibit the, so this is spindle assembly checkpoint, inhibit the assembly of the spindles if there is any error. So activation of anaphase promoting complex take place during this checkpoint and trigger the separation of the sister chromatin. And so only after proper attachment does the cell proceed to the anaphase. So the cell checks whether all chromosome are properly attached to the spindle or not. Only after that proper attachment does the cell proceed to anaphase, and this prevents the chromosome missegregation and aneuploidy. If the chromosome checkpoint fails, then DNA damage accumulates, mutations get increases, and chromosome abnormalities develop. So what will be the result of all these? So they lead to the development of disease like the cancer, and there is a, so they can, many anti-cancer drugs target cell cycle regulators come. So target of many anti-cancer drugs, importance in the diagnosis and the therapy. So this is the overview of all cell checkpoints. Now, what is the importance of the cell cycle checkpoint? So what we have learned in all these three checkpoints. So checkpoint proteins delay the cell cycle progression until the problem get repaired or fixed. If checkpoints can prevent cell division when problems cannot be fixed. And at that stage, it can induce the apoptosis if problems are so severe and cannot be repaired. And checkpoint accurately maintain the genome of the organism. Checkpoint ensure only one round replication of DNA per cell cycle. If functions of the checkpoint gene are lost due to mutation, so it will lead to the additional mutations and the cancerous growth initiate in the organ. So almost all the cancers are due to the improper functioning of either one or many proteins that involved in cell cycle regulation, that is P53 proteins. So we will learn in next lecture about the cell cycle regulations, how these checkpoints are regulated under different protein families. What are the regulatory molecules that regulate the cell cycle checkpoints?